Early Drosophila embryos consist of a continuous pool of cytoplasm in which nuclei undergo a series of rapid synchronous divisions without being separated into individual cells by cytokinesis. The nuclei disperse throughout the embryo's interior and, after 9 division cycles, most of them reach the embryonic cortex, where they anchor and eventually become enclosed by distinct plasma membranes. Little is known about how the nuclei move apart, largely because it's hard to visualize events deep inside the large yolk-packed embryo. A group of researchers, led by Ivo Telli from the EMBL in Heidelberg, therefore decided to develop a cell-free assay using cytoplasm extracted from early Drosophila embryos.
The embryos are immobilized on a glass, and then I directly go to my microscope, but I choose an embryo that is in late telophase or interphase, and I enter into the embryo and I extract the cytoplasm into the pipette, and then I move onto a clean area of the cover glass and I make little droplets of cytoplasm.
By extracting cytoplasm from embryos expressing fluorescently tagged DNA and microtubule binding proteins, Telli et al. Could observe the nuclei in these droplets undergo multiple rounds of division and dispersal, just as they would in vivo.
We tested what the cycle time is from one division to the next division, and this was remarkably similar to what is known from live embryos, but also the division itself looks perfectly fine, and the fact that it is repeating, we can observe 5 or 6 divisions, showed how robust the system is.
Telli et al. Saw that the embryonic nuclei moved apart in 2 phases. Chromosomes separated rapidly during anaphase, a process driven by elongation of the central spindle. But the daughter nuclei continued to move apart, albeit more slowly, in telophase and interphase, as the central spindle disassembled and the microtubule asters around each centrosome grew larger. In fact, the nuclei kept moving until they were separated by a distance of about 28 microns.
If a droplet has 1, 2, 4, 8 nuclei, the separation distance is roughly constant.
This 28 micron separation distance appears to be a set characteristic of the early Drosophila embryo, because the nuclei and the mitotic apparatus couldn't adapt when the researchers confined the cytoplasmic droplets in small chambers to restrict the available space.
So in the chamber experiment we created severe crowding, and that showed us that nuclei need their own space, and as soon as this is not given, they come unnaturally close to each other, and then the spindle morphology starts to be disturbed and the spindle apparatus starts to fuse.
The central spindle separates chromosomes during anaphase, but to see which part of the spindle might move nuclei during telophase and interphase, Telli et al. Treated their extracts with low doses of microtubule depolymerizing drugs, which selectively perturbed the microtubule asters at each spindle pole.
The nuclear separation is strongly reduced if we use these drugs. And then to have a more clear insight, we used UV laser ablation to locally destroy either the central spindle or 1 or 2 asters. And these experiments showed that the central spindle during telophase and interphase is dispensable, but destroying the aster then abolished the migration of the daughter nucleus. So that gave us a clear picture then that the aster was really the key for nuclear movement during telophase and interphase.
But how do the asters themselves move through the cytoplasm? In the extracts, there's no cortex for the microtubules to pull against, and in vivo, most asters would be too far away from the embryo's periphery. Telli et al. Looked at the actin cytoskeleton in their droplets and found that actin was concentrated around the mitotic spindle from metaphase to telophase.
We also interfered with actin turnover in our extract, and nuclear separation is reduced. And this now suggests 2 things. Either actin plays as an anchoring network, and the microtubule asters can move relative to this network via molecular motors, or it could be that actin is not just a static anchor, but it is actually the vehicle on which the microtubule asters are transported, and this could happen via constant turnover of actin polymer. But this is purely speculative, and further studies have to be done.
Therefore, after the initial segregation of chromosomes in anaphase, microtubule asters work with the actin cytoskeleton to move the embryonic nuclei 28 microns apart from each other. A perfect distance for the Drosophila embryo, as, after 9 divisions, the embryo interior would be filled and most nuclei would be close to the cortex ready for cellularization. But what about other insect species, whose embryos are larger or smaller, but which follow the same developmental process as Drosophila embryos? Is their nuclear separation distance altered, or do their nuclei still move roughly 28 microns apart, but undergo different numbers of divisions in order to fill their embryonic cytoplasm?
We went through the literature, and the number of cycles necessary for nuclei to migrate to the cortex varies with size. So in a small embryo, the nuclei migrate to the cortex after cycle 6 or 7, and in larger embryos, for example, from the housefly, this migration occurs after 11 divisions.
Moreover, comparing embryo size with the number of nuclear divisions suggests that a nuclear separation distance of 28 to 30 microns is conserved across most insect species. It remains to be seen what sets this distance, and Telly et al.'s study raises many other questions as well.
I think the most important question now is to find out the molecular basis of the astral migration. But I think it's important to find out if and what microtubule-based molecular motor or actin-based molecular motor is involved. And I think it's also key to find out how the two cytoskeletal networks are connected to each other with linker proteins.
Telly et al.'s cell-free assay should help to answer these questions. But in the meantime, you can learn more about the separation of nuclei in early Drosophila embryos in the paper by Telly et al., published in the June 25, 2012 edition of the Journal of Cell Biology.